Hello all.
Hello.
Hi, how's it going?
It's going okay.
Okay. If anyone needs to, chat privately with me about anything that's going on with them, then let me know. Again, so it's been a weird time, so if there are things going on and you need any assistance, there's always my email box is open, as well as I do have a Google Voice number you can call, and if it's outside class hours, I usually answer. All right. Well, we'll get started as soon as the people pop into the room. Talking about gene expression. All right. Well, I did go through all of the class assignments I have assigned so far. I have not gotten to I've not gotten any of the tests yet, so those will come up soon. Okay, get those tests as soon as I can. Okay, K Broa, who is this here? Oh, it's Kim. Kim.
Yeah.
I was like, K Bro. Okay, Kim.
Yeah.
Got it. Took me a minute. All right. My daughter's having a vigorous argument with the babysitter about whether or not she can watch another show. Hopefully that won't come through the mic too much. All right. We'll pop in the next few minutes. Oh, hopefully you guys have been having some chance to reduce your stress. I've been trying to take walks, but then we have random thunderstorms like this, so it's harder to take a walk every day. But
Random thunderstorms, jeez.
I know. It's like every time we have a reasonable day, it's like, oh, it's warm for like an hour, and now there's thunderstorms. I wish it was warmer for longer and calmer. It's starting to get into the tumultuous season.
In my local area, it isn't that drastic for weather, but it's not consistent enough for my liking.
I know.
If it was just a few days of rain, right, and it says just oh, it's raining one day, and then it's just cold with wind chill the next, and then it's just warm.
I'd like spring weather. Right now we're seeing winter weather.
Yeah.
This is the temperatures we had in November. I would like actual spring warming up weather to happen. Tomorrow will be nice but cold, and then it'll be raining for the next few days. I've been trying to put in cucumbers. They have failed to put in cucumbers. It's been either too wet or too cold to put things in the ground. So I should just start growing them in an egg carton. Get them at least ready to go. Maybe I'll have some time to put them in later. Yeah. All right. Well, we're getting more people in. That's good. So things are coming up fast, obviously, for your projects, your research projects. While we're here, does anyone have questions about research projects and finding sources or struggling with structure of the paper? How are you guys doing? Just remember, you have about a week and a half So hopefully you've done your research already, or you're doing your research actively. Give yourself at least 1 full week to write. It is due before the final, so you can, all right. So the information about the formatting of the paper and structure, all of that is in, the structure of all of that is in the instructions online. There's a outline of the paper and how to write it, all the pieces put together. You basically want about 10 sources, and none of them should be, you want at least some of them to be primary sources. You try to find actual scientific studies on this. You can do the secondary sources, but I, try not to have too many of them be like just what is a gene kind of informative sources. I want more of like write-up sources, things that are writing about scientific studies or scientific studies. So if you use something for a journal, you can probably use it in your assignment. I don't have the same requirements for your sources in your research paper in terms of the recentness of them. But if you're talking about a cutting edge thing, then it is good to have a cutting edge, article. So you can use news articles that you've used in your journals and put them together into the paper formatting. Sorry, put them together into that. The key is, again, I would like more scientific sources and sources about studies than I would just like, here is a educational resource on what, or definition of this, okay? Try not to use Encyclopedia Britannica. You want more actual written sources that describe things. Okay. I can't tell if my daughter is upset at the babysitter or if she is actually having fun screaming. Hopefully it's the latter. We'll find out. Potentially, my husband will be bothered first. Okay. So again, if you have more questions about that, realize, it is due reasonably soon. I have given you guys feedback on all of your topics. Just again, make sure that you're focusing on the genetic pieces, not just the medical pieces, especially if you're covering a disease. You should be talking about the medical parts of the disease through a genetic lens. A lot of the stuff we've been talking about, how does this protein produce this trait, but also, what is the actual error? How is it inherited? Is it inherited? What is the mutation? Different pieces like that, okay? Is it chromosomal or a gene mutation? How does gene mutation work? So, those kind of focus in, and also understanding, since we have been talking a lot about the gene therapies and treatments targeting the genes themselves, or the mRNA, or the protein products, identifying those pieces and putting them into, especially several people did diseases, so identifying how diseases, focusing on the genetic piece of it, can help to treat disease instead of just treating the symptoms. Okay. Again, send me any questions through email if you have specific questions about your paper. If you'd like me to look at a few parts of it, I can. I can at least give you feedback on you're on the right track, expand your discussion, something like that, okay? But if you want, I'm not going to give you a grade if you send it in early, but I can tell you, yes, that's good. No, you really need to focus more on this. At least give you guidance if you'd like me to. It'll just be a very broad overview, though, of what you need to fix. Okay. Again, all that paper formatting is in the research paper folder. Okay. Let's start our discussion. I will share my screen over here on our case study. Leave my chat over here. All right. And pull this over to. Okay. Get this up. All right. Okay, so this is the case study. Note that your actual assignment is a little bit less complex, but I do want us to look at some of the sources that it talks about. I linked them more directly than it is in the PDF. I linked them in the assignment. Again, if you haven't looked at this, I understand that the due date's not till tomorrow, so if you haven't looked at it yet, well, you can learn along with us. But this is about the complexity of genetic disease, in particular cystic fibrosis. Cystic fibrosis was used in a couple parts when I was discussing overall recessive genes. And it is a recessive gene, but it's not just one gene mutation. So we have to talk a little bit about more of the complexities here, okay? All right. The basics here in this meeting is that Sarah and Mike both have siblings with cystic fibrosis. And both sets of their parents did not display cystic fibrosis, indicating that they were both carriers. But carriers of what? So that's the question here, okay? And they don't have cystic fibrosis either, so Sarah and Michael. All right. So you don't have to, so when you turn in your assignment, again, you please try to draw and take a pedigree, or you can draw it in somewhere, but do try to submit that as part of it. You can submit more than one assignment file. But let's talk a little bit about the pedigree. So this one should be easy, right? I'm not asking you to draw a complex inheritance here, but let's just talk about their parents. So if we were to talk about the very basics of this recessive linked disease, remember, cystic fibrosis is the loss of function disease. And by loss of function, generally those are recessive. And the loss of function means that it does not produce a functional version of the cystic fibrosis, the specific gene. The specific protein, which is a chloride pump to clear mucus out of the lungs. So, who can tell me what, just basics, if you were to draw a Punnett square, so first we'll start with we'll get to the Punnett squares in a minute. But first, let's start with Sarah and Michael. What is their parents, and then their siblings, and then their chances of having the same mutation, or chances of carrying the allele. So you can tell me that.
Well,
plus,
since what? Oh, sorry. Testing. Okay.
I got you. I can hear you.
Okay, so for like Sarah and Michael, like both of their parents, like they'd be like carriers. So for like, in order for Sarah and Michael to for either one of them to be a carrier, like they need it'd be a 50% chance for them to be a carrier.
Because both of their parents are carriers.
Yeah.
And since they don't have it, there's only a 25% chance that they are both not carriers.
Mm-hmm.
Sorry, a 25% chance that either one of them is not carriers.
Yeah. Yeah.
Okay. So if we used So the parents are, what is their genotype for the CTFR gene? The one that produces the protein that is a chloride pump. What
they'd be
parents of
heterozygous.
either one? Yes. So all their parents are heterozygous. Okay. Parents of both are going to be big C, little c. Okay. Siblings with it, little c, and the
I guess you could call them spouses. I don't know.
Right. So the parents and the siblings, and then either one of them has a 25% chance of having it, because two heterozygotes have a 50% chance of creating another heterozygote. Okay. Chance of being a carrier. So, sorry, chance of being a carrier is 50%. Chance of both being carriers. What's the chance of both of them being carriers? If both of their parents are carriers, and they each have an individual chance of them both being carriers for an error on the CTFR gene.
Wouldn't that be, since it's an and type of probability, it'd be 25%, since it'd be 1/2 times 1/2.
Yes, it'd be 25%. Okay. This is the simple part. We learned about this early, right? We're going to talk more about the complexities now that we're further along in the class. Hopefully you guys will follow along here. Okay. All right. So in part 2, Punnett squares, this is the why doesn't the simple one work, but this is what Sarah was thinking. So we find out that Mike is the carrier of the most common disease allele, Delta F508, and Sarah is negative for the most common one. So what does Sarah think? She thinks it's safe. Now, why is it though? So we are going to talk about, it's named not exactly, okay? If we are just going by the classic Mendelian mutation concept, the classic Mendelian allele concept, let's fill in the Punnett square here. So someone else want to help me fill in the Punnett square? So let's say that this is I guess this could be bigger, but Sarah and Michael's assumed genotypes. If only Michael is the carrier, what is Sarah assuming her genotype is? So we find out that Michael.
I would. Oh,
okay.
You can go ahead. Sorry, I don't want to monopolize speaking.
Okay. All right.
Will Michael be big C, little c? Then Sarah will be 2 big C's because she doesn't have the disease.
Right. If she doesn't have the most common mutation, then she's assuming that she's big C, big C. So, so now we have, what are the 4 possibilities then if she's big C, big C?
So you have CC.
Sorry, we have a big C option.
Okay. And big C, little c. And big C, and big C, little c.
There you go. So she's saying we can only have children who are homozygous dominant or heterozygous, right? So I'm safe. Now the next question is really the crux of this case study. Who can tell me why the counselor said not exactly? Why is this not the end of the question?
Oh, well, because mutations could occur that could complicate it further than just crossing between someone who is a carrier and someone who isn't a carrier.
Now, hold on. But the key here is that carriers, what they tested for was the most common mutations. But what did we learn before about how mutation works? Can you have the same phenotype if there's two different mutations on the same gene? Okay, let's put it this way. We talked during one case study about the complementarianism, the blonde mutation and the blonde mutation creating a brown because they were in different areas of the, they were actually on different parts of the metabolic pathway, but they were still two different proteins. But what if someone was blonde and had an error on one gene was blonde and created a non-working protein in the same part of the metabolic pathway, and the other one created a no protein. Basically, they created a very truncated protein that was just wasn't present, a disappearing protein because of a nonsense mutation. Would those two still create blonde-haired children?
If they have the proteins that is missing in the metabolic pathway so that they can fill those out.
Okay, so we're talking about two different things here, I still think. Angie, did you have something to say?
Oh, I just said I don't think so.
Okay.
Also,
So let's go ahead.
were you talking? So you're saying that maybe the doctor might be saying that she might have a different type of mutation on the different metabolic pathway.
What he's saying is actually she may have a different kind of mutation than they tested for. Okay, so let me explain the difference between the two things I was saying. Okay, the complementarianism is a different concept than we're talking about. In that case, two people with the same phenotype could produce a different genotype because it's not the same allele. The key here is that mutations on the CTFR gene, multiple mutations and multiple kinds of mutations can create overall the same effect. Okay, so let me show you this. Okay, let me zoom in so everyone can see what I'm writing. All right. So here's the deal. If so the specific one that is mentioned, the Michael, has one allele that creates a working CTFR protein. That's all he needs, one allele, okay? Other allele is missing. This is the most common mutation, phenylalanine, and is non-working. Since he's producing some of the working protein, he does not have the symptoms of cystic fibrosis. Now, is that the only way that people can have a non-working protein? is that the only way that that CTFR gene can be broken is to have those missing 3, because it has a deletion mutation of 3, so it's just missing 1 amino acid. It's not a frame shift because it's 3 altogether, but it skips 1 amino acid. Is that the only way that you could make a non-working protein?
No.
How else could the protein be broken? We talked about gene mutations last class, so hopefully you guys can remember this. How else can it be
There
broken?
could be an insertion or deletion error.
So perhaps it's actually, if there's an insertion or deletion error early on, that's not 3 in a row, like the one that Michael has is 3 in a row, okay? So that is skipping only 1 amino acid. But if it's a skipping of 1, you could have a completely nonsense, it's called missense, extensive missense frame shift, and you could create a completely non-working protein. What else could happen? So that's a different mutation than Michael has. But you could still come up with a non-working one, okay? So let's take the next one. Where else could it be broken on the gene that would make a non-working protein? What else could happen?
Can we have a nonsense mutation? So where there's like a
Yes,
stop?
a nonsense mutation.
Okay.
So it could be a point mutation that just creates, instead of making a amino acid, it makes a stop codon, and you create a truncated protein. That truncated protein could absolutely create a non-working one. Now here's the deal. Does Sarah need to have the same mutation as Michael to have a child with cystic fibrosis?
No.
Oh.
No. So let's write out what would happen within one of those scenarios. Let's say she has the nonsense mutation. Sarah has a nonsense mutation on one chromosome. Then that protein will be non-working, okay? And she has one working, just like Michael, because she doesn't have the disease. But here's the key here, is that not exactly is because they don't know what mutation her sibling has. So if you look back here, actually, you'll note this word. He had a younger brother who died. I had a younger sister. There's no way to test their siblings now for the trait. And her parents could have 2 different mutations as well. So testing them is not going to tell her whether she has she shares the same one as her brother had. So what they need to do is test Sarah, but they need to know what they test, okay? So we're next going to go through here, okay? So please, when you're doing this, pay attention. This is long, but I want to point out a few pieces here, okay? I did mention this during class last time. The 508th triplet codon, with normally codes for phenylalanine, I can never say it right. That's deleted, okay? So this is the one that he has, okay? He said F508, but it's the same thing, PHE 508, okay? It's that triplet codon, the most common mutation, okay? But let's explore these other ones, okay? So not all of them, but let's look here about in the classes, okay? So I'm gonna go click through here to this one. I'm going to highlight it and then copy it into my Safari here. Oh. Brief preview of what you're gonna see later. I was just pulling that up. All right, let's go to the window. Okay. Oh, okay. Apparently that does not make it come up. Oh, okay. There we go. Okay, so this one, the key here is that it's showing all the different domains, okay? So each one of these has a variety of domains that build the whole protein here, okay? And what this shows you is this thing. So this was included in last week's presentation about gene mutations, okay? Here are the options. Are you not going to zoom in? Okay. Not doing it. Okay, here. Okay, so the most common one is class 2, okay? The deal here is that you get the full-length RNA, but because it has an error, it is misfolded, and that misfolding causes you to not have functional CTFR. It does still go there, but it doesn't function properly. But you can see that there are these other options. This one is a unstable truncated RNA. That's that class, number 1. Unstable truncated, and that one is likely a nonsense mutation, okay? So what happens here is that this is the defective channel regulation, okay? And this one instead just creates, it does not fold properly, so it has defective channel. This one is decreased channel conductance because of an error in shape. This one is reduced synthesis. This one is reduced stability. So there's several different ones that you can have. These are just the major classifications, okay? All right. So let's go to the actual website here. Okay, it tells you to go to OMIM. So what I'd like you guys to do is we'll go through, if you can go here, again, don't, you just cut that out, it'll go straight there. Okay, here we go, cystic fibrosis, okay. So what we'll do is we are gonna go to the allelic variants, okay. I selected a few, but I want you guys to, in the background, go to this website here, okay, omim.org. You can just type in that and then type in cystic fibrosis and this will pop up. The key is this is the cystic fibrosis transmembrane conductance regulator, okay? That's the CFTR, sorry, I was saying CTFR. CFTR gene, that's the one that is creates cystic fibrosis because it's the transmembrane conductance regulator, okay? Go there, go to allelic variants, and then look up these 3, okay? So why don't you spend a few minutes doing that, looking that up on your own. If you have trouble finding them, let me know, but you know, go to 0.004, 0.0019, 0.0064. And what you want to do is you want to identify whether it's a nonsense, missense, insertion, deletion, or splicing error, which there's a bunch of them that it could be, okay? and then explain how, what I want you to do in this is to explain how it can, each of those can cause cystic fibrosis. So what I'll do actually is, let me, how about I assign it to you guys, and then you guys can go through it in pieces. Angie, Fanye, and Everett, Fanye and Everett, you're already in the same room. Why don't you go for the 0.0004? Okay.
Okay.
Mavis, Stacy, Asa, and Kim, why don't you look up that 0.0019?
All right.
And Sanusi, Faye, and Anik, everyone else, Ganyu and Dana as well. Why don't you guys look up the last one, the 0064, okay?
Okay.
You guys got your assignments?
Okay.
Yes.
Okay. All right, great. Go do that. I can't see you looking it up, but go do those. You don't have to read off of my screen, so you can actually be in charge of your own reading. Read through those, and I'll give you guys about, let's say, 6 or 7 minutes to do that. And we'll come back together and talk about the, we'll talk about these three and what causes them and how can each one of them independently cause cystic fibrosis. What do they do? Okay. So this gives, so you see for print 0001, that gives all the information about what it is and what it causes, okay? But go to the other ones to see that too.
So can I see the website again? Can you show the website?
It's omim.org. And so what you can type in /entry/60242. Okay, that's also in here. Okay, 60242. Sorry, 60241. So that's the entry for this specific gene and its mutation. And if you type #allelic variants, it'll take you straight down to that, but you can also click on the side, as I did. You will have to look on up the exons and introns yourself, but right now I think it's more useful to go through those mutations and understand how they can cause cystic fibrosis. Again, just do the, so we're going through the same three that are in my information. There's more in the actual case study. I picked a few that I thought were representative. Again, you don't have to go through all of them that are in the actual case study, but do go through these three and give explanations to them, okay? Couple more minutes. Examine, give some information here of what kind of a mutation is it, and then the details would be, this tells you, okay, what is it? This is deleted, this is shorter, one amino shorter, missing its unit. Okay, what is it? Is it, what kind of a mutation is it, okay? All right. Let's see. I guess it's been about 5 minutes, not 7. So, how are you guys doing? Need more time? Do you understand what you're doing? Have you gotten to the website? Do you understand the segments that I've assigned you?
Yeah, I have. I know it's just like the
Okay.
0.0019. Yeah, I was able to find that. Yeah.
Okay, good. So we'll go through them in a minute. Still a second to go through them, and we'll go through one by one and have the people in the groups describe the different ones that I assigned it to you, and I've already forgotten who I assigned to what, so you'll have to remember for yourself and tell me which ones you read and identified, okay? Again, listen to the other ones because these are part of your assignment, and you'll be able to type them out, okay? So let's type out, I'll type it out for you guys. Again, you'll have to do this yourself, but so who can tell me about 0.0004?
Okay, so for 0.0004, cystic fibrosis is a missense mutation. And this is because the protein aspartic acid is replaced with histidine.
Okay, so here you go. And this tells you it's a different one that's than is the one replaced in the most common mutation, that 0.0001. Still a missense, okay? So it's a C to G transversion. It replaces one with another. It's a single point mutation. There's no deletion, but it has replaced one. Okay, so this is missense, not deletion, which the other one is a deletion, okay? Note, mild cystic fibrosis, okay? So this one would not present the same way, but it still does cause cystic fibrosis. Okay. This would be missense and mild disease. Okay. What about 0.0019? Who can tell me about 0.0019? Who's in the group that I assigned there?
Yeah. For 0.0019, it's like a frame shift mutation because it inserts two nucleotides. Yeah, it inserts two nucleotides specifically for exon 13. I was trying to figure out where exon what exon 13 did. So I tried to control F and like it appears. I couldn't find anything specific on this page, but
this is a frame shift, okay? So this is the person who has all of the cystic fibrosis symptoms, indicating that that frame shift caused him to have a non-working the CFTR gene, CFTR protein, okay? So whatever it controls, so we can go back actually to the previous one here. Hold on, let me go here. This one does tell you about the various exons and introns. Directly there. There we go. Okay. There you go. So it has 27 exons. So they count from in the way that it is organized. So if there's a missense in number 13, it's somewhere around here, then everything past that is broken. It's not the correct order, okay? All right, so the answer for number 19 is, this is a frame shift mutation in middle of gene, okay? So it does shift a lot of amino acids. Remember, frame shifts are a little bit more dangerous in terms of functionality. So he has a completely non-functional one, okay. Next, for this one, 0.0064. Who wants to tell me about this?
I have the fact that it's a splice mutation at the 5' end of the neutron of intron 12. That's what I got.
Okay. 0.0064. All right. Splice mutation. That means that it's not splicing it properly. It results in skipping an exon. An exon is more than one amino acid, so this means now that this is a different piece. The spliceosome was not identifying the proper places to splice. This is a splicing error, not. So technically, they have all of the necessary nucleotides in the actual sequence, but those SNRPs, those proteins that latch on and say where to splice, they're not doing it right. This is a splicing mutation. Okay.
So, Professor, is this why Sarah has to be
Yeah.
careful? Why the doctor was like she needs to be careful? Because technically, it looks like she's normal, but she might have the 0.0064 where the splicing occurs.
Right. Well, this is the thing. It doesn't matter if she's normal. She has, she knows there's a 50% chance that her parents passed on to her one of the errors that caused her sibling to die. Okay? Something to comprehend here. Let's figure out what would happen. What would happen if Sarah had a splicing mutation and her husband had a, so he has one allele, one of his homologous chromosomes contains that broken missing phenylalanine gene, okay? And if one of hers had a splicing mutation, what would that, and she gave the splicing mutation allele, and he gave the deletion allele, what would that child end up as? Would they have cystic fibrosis?
Professor, can you repeat that again?
Okay. So let's pretend Sarah has this one. If she passed this allele to her child, and because remember, she has two alleles. One's working because she doesn't have cystic fibrosis, and one's not. She will not have the symptoms if she has the non-working one as long as she has one working. But if she passed the non-working allele, and Michael passed the FE508 error allele, would the child have cystic fibrosis?
Oh yeah, the child will have cystic fibrosis.
Because neither of those alleles create a proper protein. No matter if it's two different mutations, if the child cannot create a proper protein, if neither of the alleles are creating a proper protein, even two different mutations can cause the cystic fibrosis to happen. Whichever one of these proteins was more functional would be the one that would likely make it to there, but neither one is very functional. Okay. Now note, this missense, if she had the missense one, and that was paired with the broken one, then potentially the child would only have a mild disease and live longer. Because this one works a little bit. It's one of the other errors, one of the other classes that I showed you before. The classes of the error in working, not just absence of a protein. It doesn't work properly. But if even there was some that worked properly, the disease would be mild. So she might have more hope if she had this one versus the completely not working one that these people have, okay? All right. Okay, so we already talked about how they can all cause cystic fibrosis a little bit. Okay. So next, let's go here. I want to try to get through most of this here, but this doesn't want to open stuff from my Word, so I'm just copy it here. Okay. All right. So let's find out how many together. So here you go. 1,700 different mutations in the CFTR gene cause CS. A lot of mutations. All right. The standard test. Now note, this is something interesting about tests. If you send off your genes to be tested for a specific trait, for a specific gene mutation, do they sequence your genome? Does anyone know how exactly gene testing works?
No.
No, they don't sequence your entire genome. Do you know how it does work? So it's a form of that PCR that we talked about. Basically, it tags and copies specific segments, and it can amplify a specific segment so they can detect that you have it. Basically, it won't amplify if you don't have the tagged gene of interest. They put a bunch of that gene of interest, little snippets that tag onto something specific about that mutation, and then they replicate, and if it replicates over and over, then it has found that in your genes. But it can't do that if you don't put a tag in to find it. So let's look at how many mutations the standard test detects. So, could Sarah have a different mutation than is tested for?
Yes, she could.
I'm sorry, Kim, I'm having trouble hearing you. It's kind of
Yeah.
buzzing when you turn it on.
Can you hear me now?
Sorry. Can you type it in the chat? Because I can't actually hear you. Maybe an issue with your microphone. So can Sarah have a one of the ones that was not tested for in the test?
Yeah.
I'm having trouble hearing anyone. Maybe I'm having a connection issue. Not sure. If you guys can still hear me, that's good. But anyways, the answer is yes. She could still have a mutation. Okay, she could still have a different mutation because there's 1,700 that cause it. They only test for the most common ones. Since she has a sibling and she's so high risk to have it, 50% chance that she has it, it's likely she should have a more comprehensive genome sequencing test. What they do is they actually sequence the gene of interest and find out if there are any errors. Okay. All right. So we already talked about this. 88% of the carriers are detected overall, okay? All right. So Sarah finds out she carries one of the less common mutations. This happens in this last part here. Okay? Last part, not the test, but here. Okay. So, it's a lot of information here. But here you go. DNA sequence indicates that Sarah has a single base pair difference about 100 bases before the ATG start codon of the CFTR gene. I clarify that for you because I know it's a lot to interpret. But it's upstream of the start codon sequence. Now note, the start codon isn't the beginning of RNA transcription. What is transcribed before the start codon? Who can tell me? Who remembers what's part of your RNA transcript?
Is it the promoter?
Okay, so someone said promoter, I think. I'm having some trouble with my actual hearing of the information. Promoter. Yes, the promoter is also transcribed. So if Sarah has a promoter error, what could that do to the protein? What could that do to the mRNA and then the protein? What would happen if you had a error on the protein?
Possibly not having that gene transcribed, thus not having it like translated to a protein.
Right. Still having trouble hearing people, but I heard most of that. So so Angie said that potentially that gene will just not be transcribed. That because the promoter is where the RNA polymerase is identified to transcribe. So she has a different one, and actually promoter mutations. We're going to talk about this actually next on Thursday. I hadn't gotten to it in my description here, but promoter mutations, actually some of the worst kind of mutations because they stop the transcription overall, missing proteins, not just truncated ones, but absent. This is a lost protein. That's no good, okay? So if there were 2 copies of this, then the person just wouldn't have this transmembrane conductance protein at all. And that would obviously cause cystic fibrosis. All right, the last part is more kind of discussion here. What would you do if you found out this was true? Again, the same options would be if you found out that you both carried the sickle cell allele. This is a similar thing that would be presented to you by a genetic counselor. So, again, there are these options here. All right. And this is a good question. Some people, the question is, you know, they have a 25% chance of having, so they have a 75% chance of having a normal child, even with both of their mutations. So was it better to do it this way? Good question. All right. So let me put this up for a minute, and this will be the last thing. Again, I'd like you to explain this a little bit more in detail during your actual thing, okay? So explain a little bit about how having those 2 different mutations could still end up with a CF child. Explain how the promoter can actually impact that, okay? So obviously, if you're here listening to me talk, you know the answer to that, but explain it. And then also, but I'd like you to guys share about, you know, what if you found out that, you know, you had a mutation that had a 25% chance of your child having a fatal disease? What would you choose if you wanted to have biological children? So here are the options that were given to them. Anyone have any thoughts about that? There are options. Options aren't that great, but there are options.
They could do the genetic testing. What is that, option D?
So this one would be get pregnant, test the embryo via the chronic villus sampling, and then terminate if they got that 25%.
Oh, well, that's kind of sad. But I think that the testing could be a positive thing, right? Because, I mean, it would give the mother reassurance if it's negative. But if it's positive, then I guess that would be more of like an ethical thing, whether they decide they'd want to terminate. But, you know.
That would be a difficult decision, right? Question is whether, so some people, you know, roll the dice and they say, you know, let's test. Some people, they decide to do the D and they end up going through with C because they hope that there will be, maybe this child will have a milder form of cystic fibrosis. Perhaps there's treatments. They can't go through with D. So sometimes that happens, even if they know that the embryo has it. So they don't always follow through to the last part. It's complicated when you're going through it, right?
Indeed.
Say again?
No, I was just agreeing, that's all. I wasn't adding anything.
Yeah. Obviously, A and B have complex feelings associated with them as well, especially if you're trying to have a child.
I feel like E could also be a good option just because of there's been a lot of like positive results for women who have done like in vitro fertilization. Because I mean, that way they could get their eggs screened first before implantation, even though that's people still
It
question.
still has ethical implications here, is that if you end up creating embryos that do have the error, you're not pregnant with them, so you don't have to go through a induced abortion.
Right.
But they are still created, and so that there's that ethical piece of, you know, where is the okay to say? And note that people who go through IVF often have screening done in order to identify not just the cystic fibrosis, but they go through screening in particular to identify that they have the whether or not there's a chromosomal error. Because chromosomal errors do happen reasonably often, the screening often ends up finding out that 50% of the embryos that were created would not be viable in the womb. And so that can be difficult for people, but that's sort of with cystic fibrosis is complicated because the kid would be born and be able to live until 15 or 16 and then pass away, which is a difficult thing to decide that for an embryo as well. Tough, right? These are some of the genetic things that are good to wrestle with, to consider that we have technology. That if they didn't know that they could be carriers, then they may even go on to have 2 or 3 children that don't have them, and not know that they had that risk. And this is the complication of genetic testing. Is knowledge good?
I would like to say that like it is at least good to have some information. Like I know it can be like, you know, like a downer like when reality kind of hits, but like it's better to at least be less blind about it, if that makes sense. Like if it's something
Right.
that like if it's something that like if you know like there's like a family history by like and you're somewhat curious about like I think the person kind of owes it to themselves to like kind of like at least and investigate on it. Well, how rigorous
Right.
they want to go into that, I can't really say it, but you know, just having some curiosity wouldn't be like I think they should have some of that.
Right. Yeah, and knowledge is generally good, especially when you're preparing for a child. You know, I know that for there. So there's people who are like, oh, I don't want to get genetic testing. Like older women say, I don't want to get genetic testing and find out about Down syndrome because I would keep the child no matter what. So here's the deal, though, is that it doesn't have to be a if you test, you terminate. It can be a choosing to keep the child, especially if there's an accidental pregnancy and then they decide to keep the child. That is still a personal decision, even if they know the child will have major issues. But being prepared is good, right?
Right.
Yeah. I will note also that, you know, all of these, they have issues, right? There's no simple answers for this couple, and that's why they go through genetic counseling, because they need to know what their options are, and they need to know what the implications of the options are. I know that some people will not get testing for something like if they know they have a chance of having dementia. They won't get testing for it because it's already decided for them. So they choose not to know unless they show symptoms. And it's an interesting sort of like people who don't want to get tested for like genes that contribute to breast cancer, because that would just cause them undue stress. They don't actually even know if they would get breast cancer. So this is a complex piece, especially for personal testing versus testing of a child that would, you know, would have an entire life of potential suffering ahead of them. So it's a little bit different for personal, like maybe Huntington's or familial dementia choosing not to. Anyone else have comments about this part? Again, please write out your thoughts. And you're graded on doing both the parts of describing the mutations and doing and talking about how promoter error can cause that. But also, you were in charge of writing out the, like, you write out your thoughts about testing and choosing testing, and then also about what would you choose in this situation? All right. And well, if no one has any more comments, then we're done. But again, you can write out everything that I just said tonight. That's fine. It's, I won't count this as plagiarism if you type out something that I said. But again, do go back and read those sources and make sure that you can describe them clearly what is going on with those different mutations. If on the final I describe a mutation, you should be able to tell me what is the mutation. So if I described this person's missing 3 sequential nucleotides, well, what would that cause in the actual gene? What would that be called? This person has an insertion of 2 nucleotides. What would that cause? Those kind of things I think are important to follow here. All right. Again, if you have, if you do have any more questions, you can ask me. Feel free to message me privately right now if you want to, and can continue on. Again, if you have more questions, let me know. Okay, everyone have a great day. I'm going to stay in here for a few more minutes. I think a couple of people want to chat, but figure that out.
All right. You all have a good day now.
Okay, bye.
Bye, Professor. Bye, everyone.
Bye.
Bye, guys.
Professor, I messaged you privately. I don't know if you have.
Do you want to chat right now? We'd probably be better to go into a different chat room, but I can send you the link to get into a different chat room with me. Would that be good?
Sure. Thank you.
Okay. Sounds good. Give me about 5 minutes. So let me send you the link to my personal chat room for here.
Thanks.
Okay.
Is it gonna come through?
Yeah, I'll come
message?
to the private text, the private chat right here. So there, you should have it now.
Okay.
Okay. All right. Does anyone else want to chat? I can set it up so I can chat with one person or another, but anyone else? No? Okay. All right. See you guys later.